the whole game.
Right. So the question that it kept me up reading through this material is, why doesn't it kill us? How does this tiny chemical know to blow up the bacteria but leave your liver and your brain cells alone? Because to a layperson, a cell is a cell.
It is. And the answer seems simple on the surface. People just say, oh, it's targeted. But when you actually peel back the layers, which is what we're doing today with this source, Microbiome, you realize that safety is, well, it's an illusion. Or rather, it's a very fragile truce.
That is a slightly ominous way to start, a fragile truce.
It is, because we are walking a very, very fine line every time we prescribe these things. The gap between helping and hurting can be razor thin.
Well, that is what we are digging into today. This source, Microbiome, it covers antimicrobials, antivirals, and immunomodulation. But let me tell you, this isn't just some list of drugs. It reads more like a manual for a bomb disposal unit.
It really does. I had the same thought, because it is specifically focusing on the central nervous system, the CNS, the brain. And treating infections in the brain is, it's a nightmare scenario for any physician. You're trying to conduct warfare inside a very delicate, closed room.
Right.
If you set off a grenade in an open field, it's one thing. If you set off that same grenade in a china shop,
Bad things happen.
very bad things. You might kill the bad guy, but you destroy everything else in the process.
So we're going to look at what this source calls the drug target map. We want to understand the hidden dangers of treating these infections, and we are going to uncover some serious collateral damage that I guarantee you haven't thought about. I certainly hadn't.
Specifically, how these very common drugs might be messing with your mood and your energy levels long, long after the infection is gone.
Okay, let's unpack this. The source starts with a core principle. It's almost philosophical, which I found really interesting. It says, antimicrobials do not work because they are strong. They work because they exploit biological asymmetries.
That is the orienting truth for everything we are going to talk about today. Biological asymmetry. If you get one thing, get that.
Biological asymmetry. It sounds elegant, but it feels a bit abstract. Unpack that for me. What does that actually mean?
It's the orienting truth of the whole text. And honestly, it's the only reason we survived the 20th century. Think about it this way. Warfare usually involves shooting something, right?
Right.
But in medicine, the battlefield is you. You are the terrain. The hostages and the terrorists are all mixed together in the same room.
Okay, so I'm the crowded room where the fight is happening.
Exactly. So if I want to shoot a bad guy who's standing in the middle of a crowd of hostages, I can't use a grenade. I can't even use a shotgun.
You need a sniper rifle.
You need a sniper rifle. And more importantly, I need a way to tell the bad guy apart from the hostages. I need to know the bad guy is wearing, a bright red hat, and none of the hostages are.
Okay. The red hat.
That red hat, that's the asymmetry. You have to find a biological feature, a specific protein, a cell wall, an enzyme, that the germ has effectively painted in bright neon orange, and that the human host doesn't have at all.
Because if I have it too, if I'm also wearing a red hat,
then the bullet hits you too. That's the fundamental rule Microbiome lays out, and it's so important. The more similar the invader is to the host, the fewer safe drug targets exist.
This makes so much sense. It immediately explains why antibiotics are generally safer than, say, chemotherapy.
Precisely. Think about chemotherapy for a second. Why is it so brutal? Why do people lose their hair and get so sick?
Because cancer cells are our own cells. They've gone rogue.
Correct. They are human cells. They're just growing out of control. So finding a difference in asymmetry, that red hat between a cancer cell and a healthy cell, is incredibly hard.
There's not much of one.
Not much at all. You're mostly targeting fast growth. But what else in your body grows fast?
Your hair, your gut lining.
Your gut lining, your bone marrow. So the drug attacks them all. It's not a sniper rifle. It's more like a cluster bomb. It causes a ton of collateral damage.
Whereas bacteria, they are completely different.
Bacteria are prokaryotes. They are very different from us. They split off from our evolutionary tree billions of years ago. They are like alien life forms compared to human cells.
So there are a lot of red hats to aim for.
A lot of big, obvious ones. But, and this is the big but that the source really drills down on, when you get to fungi or parasites.
Those are eukaryotes. They're the same big family as us.
They are our cousins. Their cells look a lot like our cells. They have a nucleus, they have mitochondria, all the same basic machinery. So finding a poison that kills them but not us becomes incredibly, incredibly difficult. The red hats get smaller and smaller.
Okay, so let's get into this drug target map that the source outlines. It breaks it down by pathogen, bacteria, fungi, and viruses. Let's start with what we think of as the safe zone, bacteria.
Right, so looking at the map, what does a bacterium have that you don't? If you were zooming in with an electron microscope, what is the first thing you'd bump into?
The text mentions the cell wall, something called peptidoglycan.
Peptidoglycan. It's this rigid, mesh-like armor. Think of it like chainmail wrapped around the bacterium.
And humans are squishy. We don't have that.
We are very squishy. Our cells have membranes, which are more like soap bubbles, flexible, fluid. But bacteria, they're under immense internal pressure.
What do you mean, pressure?
Osmotic pressure. There's so much stuff packed inside them that water is constantly trying to rush in and swell them up. It's like a car tire pumped up to 50 psi.
Yeah.
If they didn't have that rigid peptidoglycan wall to hold it all in, they would literally just pop, explode.
So the asymmetry is they have a wall, we have a bubble.
Exactly. So if we design a drug that targets the wall, if we stop them from building it or maintaining it, we are hitting a structure that simply doesn't exist in human cells. That is a massive, beautiful asymmetry. It's the safest target we have.
Okay, check. A nice big red hat. What else?
Ribosomes, the machinery inside the cell that reads the genetic code and makes proteins.
But wait, hold on. Humans have ribosomes too. We need to make proteins to live. I remember this from biology class. If we stop protein synthesis, we die.
We do. We absolutely do. But the text highlights a crucial difference in the numbers. It's subtle, but it's the kind of subtlety that saves your life. Bacteria have what are called 70S ribosomes. Humans have 80S ribosomes.
The S is just a measure of size and shape.
It's a measure of how they sediment in a centrifuge. But yeah, for our purposes, think of it as a difference in their size, shape, and composition. It's like the difference between two models of an engine.
Okay.
Think of the bacterial 70S ribosome as a diesel engine and the human 80S ribosome as a high-tech electric motor. They both make the car move, they both make proteins, but the parts are different. The spark plugs are a different size, the filters are a different shape.
So you can design a tool for one that won't fit the other.
Exactly. You can throw a wrench specifically designed for the diesel engine, that's the antibiotic, and it just won't fit into the electric motor of the human cell.
Theoretical.
Theoretically, the ADS engine keeps running smooth, usually.
Usually. I feel like we're going to come back to that usually, because I have a feeling that's where the collateral damage comes in.
You are right to be suspicious. Evolution is messy. Nothing is ever 100% perfectly distinct. There are always overlaps.
Okay, so that's bacteria. Pretty different. Now let's look at fungi. The source says the difference here is much smaller. We're moving from aliens to cousins.
Much, much smaller. Fungi are eukaryotes, just like us. So they don't have that weird peptidoglycan wall. They have membranes, just like our cells.
So how do we kill them if we can't target a wall?
We have to get much more specific. We look at the fat, the lipid composition of that membrane.
The fat.
Yep. Fungi use a specific type of steroid molecule called ergosterol to keep their membranes stable and fluid.
And humans.
We use cholesterol.
Ergosterol versus cholesterol.
Yeah.
That sounds suspiciously similar.
It is. It's a chemical game of inches. If
Yeah.
you look at the molecules side by side, they are almost identical. There's just a tiny difference in a side chain.
So the red hat is more like a slightly different shade of pink hat.
A perfect analogy. So we have drugs that are designed to attack ergosterol, but because the molecules are so similar to our own cholesterol, there is a very high risk of cross-reaction.
The drug gets confused.
Essentially, it attacks the fungal membrane, which is what we want, but it might also take a swipe at your own cell membranes in your liver or your kidneys or your brain.
And that's why antifungal medications can be so toxic.
That's exactly why. That's why we have to monitor liver function so closely when people are on them. The asymmetry is weak. The target is just too close to home.
Okay. And then we have viruses. The map says they use things like viral polymerases or reverse transcriptase. What does that mean?
Right. Viruses are the ultimate minimalists. They're ghosts. They don't even have their own cells, really. They are just a bit of genetic code and a protein code.
That hijack our machinery.
They hijack everything, so we can't target a viral cell. We have to target the specific, unique tools they bring with them into our cells. Think of it like a burglar who brings his own special lock-picking kit. We can't attack the burglar directly, but maybe we can design something that breaks his tools.
Okay, so what are the tools?
Things like a specific enzyme that copies their genetic code, a viral polymerase. Or in the case of retroviruses like HIV, an enzyme called reverse transcriptase, which does something very unnatural.
It turns RNA back into DNA.
Exactly. Human cells don't do that. So that's a great target. If we can block that enzyme, we stop the virus in its tracks without messing with our own cellular processes.
So that's the landscape. We have these targets. Now, I want to get into the specific weapons we use. The source breaks antibiotics down into 4 major groups. And this is where we get into the nitty-gritty of how these things actually work and, more importantly, how they can go wrong. Group A is the cell wall synthesis inhibitors.
These are your heavy hitters, the classics. Penicillins, all the beta-lactams, glycopeptides like vancomycin. These are the drugs that target that peptidoglycan wall we talked about.
How do they work? Walk me through the mechanics of it.
Okay, so imagine the bacteria is constantly trying to build and reinforce that brick wall around itself, right?
Yeah.
It's laying down bricks of peptidoglycan.
Right.
But just stacking them isn't enough. You need mortar to hold them together. You need to cross-link them to make the wall strong. There's a special enzyme that does that applies the mortar.
Okay.
These drugs, the beta-lactams, they are shaped perfectly to fit into that enzyme and block it. They gum up the works. They steal the trowel.
So the bacteria keep stacking bricks.
But they aren't glued together.
All right.
And remember what we said about that massive internal pressure.
That's 50 psi.
Right. So
Yeah.
the bacteria is trying to grow. It's pushing out against this wall. But the wall is now weak. It has no structural integrity.
So buckles.
It doesn't just buckle. It catastrophically fails. The internal membrane starts to bulge out through the weak spots in the wall and pop.
Osmotic lysis.
Osmotic lysis. The bacteria literally explodes from its own internal pressure.
Now, here is where it gets really interesting and honestly kind of terrifying for our listeners concerned about the brain. The source has a special neuronote here. It says the drug itself isn't directly toxic to the brain. But
the explosion is.
that is such a vivid and unsettling image.
Think about it. You are in the brain. It is a closed box. You have the skull, which doesn't expand. You have a serious infection like meningitis. So you throw in a powerful drug, say ceftriaxone, that causes millions, maybe billions of bacteria to explode all at once. What happens next?
Debris everywhere. Bacterial shrapnel.
Exactly. You release all that debris chunks of cell wall, toxins, internal bacterial gut. This is what we call endotoxin or peptidoglycan fragments.
And the immune system sees that debris.
And it goes absolutely crazy. It screams, "Invasion, code red," and launches a massive, overwhelming inflammatory response. It sends in fluid, white blood cells, cytokines, all the tools of war.
But inside the skull, there's no room for extra fluid. There's no room for swelling.
None. So you get cerebral edema brain swelling. The pressure inside the skull skyrockets. In the medical world, we see this all the time. You give the right antibiotic to cure the meningitis, and for the first 12 to 24 hours, the patient actually gets worse.
Worse.
They might slip into a deeper coma, their fever might spike, they might have a seizure.
Because you're killing the bug, but you're effectively setting off a grenade in a crowded room.
That is a perfect analogy. You are winning the battle against the bug, but the shockwave from your victory is what's damaging the host. It's a paradox of treatment.
That brings a whole new meaning to it gets worse before it gets better.
In CNS infections, that is literally the clinical course we expect and have to manage.
Okay, let's move to Group B. This is where I had my biggest aha moment reading this source. Protein synthesis inhibitors. These are the ones that target that 70s ribosome we mentioned.
Right. Drugs like tetracyclines, macrolides, aminoglycosides. That job is to gum up the gears of the bacterial protein factory so it can't produce the proteins it needs to survive.
And we said this is generally safe because humans have ADS ribosomes. They have the electric motor, they have the diesel, but the source has a neurorelevance warning here that connects to evolution. And this blew my mind.
Yes. This is one of the most fascinating and, I think, underappreciated concepts in pharmacology. Do you remember learning about mitochondria in school?
The powerhouse of the cell.
The powerhouse of the cell. The meme, right.
Of course.
Well, where did our mitochondria come from? Why did they have their own separate DNA?
I don't think I ever learned that part.
Evolutionary biology, through something called the endosymbiotic theory, tells us that billions of years ago, mitochondria were likely free-living bacteria.
They were their own independent little bug.
Exactly. They were just floating around. And one day, a larger, more complex cell swallowed one of these little bacteria, but instead of digesting it, they formed a partnership.
A deal?
They made a deal. The little bacteria basically said, "Hey, if you protect me and give me nutrients, I'll use oxygen to make a ton of energy for you."
The ultimate roommate agreement.
Precisely. They became endosymbionts. And now they are ancient bacteria living inside every single one of your cells right now, powering your body.
Okay, so they are ancient bacteria. What does that have to do with antibiotics?
Well, because they are effectively bacteria, guess what kind of ribosomes they have?
Yeah, don't tell me it's not ADS, is it?
They look a lot more like bacterial 70S ribosomes than our own ADS
Oh,
ones.
wow. Okay, I see the problem.
So, when you take an antibiotic like a linezolid or a tetracycline that is specifically designed to target the bacterial ribosome,
It hunts down the strep throat infection.
but it also drifts into your own human cells, finds your mitochondria, sees that 70S-like ribosome and thinks, aha, an enemy.
And it jams the wrench in the gears of your own power plant.
It creates off-target effects. The drug thinks it's attacking an invader, but it's actually attacking the energy source of your own body. It's friendly fire on a microscopic scale.
That is wild. So I'm taking a pill to get better, and chemically, I'm sabotaging my own energy production.
You are. And the source lists the specific consequences of this mitochondrial damage. It's not just fatigue. It mentions neuropathy, which is nerve damage, and ototoxicity, hearing damage.
Hearing damage from an antibiotic. Why the ears specifically?
Think about the cells in your inner ear. The tiny hair cells that detect sound waves and turn them into nerve signals, they are incredibly metabolically active.
They're always working.
Always firing. They are packed, absolutely packed with mitochondria to meet that energy demand.
So they are more vulnerable to a power outage.
Exactly. They're like a city's hospital or data center. They need constant, reliable power. If you damage those mitochondria with certain antibiotics, aminoglycosides are notorious for this, the hair cells starve for energy and they die.
And they don't grow back.
They don't grow back.
Yeah.
You can permanently lose your hearing or develop severe tinnitus from a course of antibiotics.
That completely changes how I view these drugs. It's not just may cause upset stomach. It's may cause a siege on your mitochondria.
It helps explain why people feel so profoundly wiped out when they are on strong, broad-spectrum antibiotics. It might not just be the infection making you tired. Your mitochondria are literally struggling to breathe.
Incredible.
Yeah.
Okay, let's briefly touch on group C nucleic acid synthesis inhibitors. These stop DNA replication.
These are your fluoroquinolones like ciprofloxacin. The CNS caution here in the source is about lowering the seizure threshold.
Right, it says they can lower the seizure threshold or cause confusion, even psychosis in some cases. How does blocking bacterial DNA lead to a seizure in a human brain?
It's a case of mistaken identity again, molecular mimicry. These drugs target an enzyme in bacteria called DNA gyrase. Gyrase is like a disentangler for DNA. It unties the knots in the DNA double helix so it can be copied.
Okay.
But the drug molecule, just its physical shape, looks a little bit like the key that fits into a certain receptor in the human brain, the GABA receptor.
GABA, that's the main inhibitory neurotransmitter, right? The one that calms the brain down.
Correct. GABA is the brain's brake pedal. It keeps your neurons from firing too much, from getting overexcited.
Yeah.
Some of these antibiotics, like Cipro, can accidentally bind to and block that GABA receptor.
So you cut the brake lines.
To cut the brake lines. Suddenly, there's nothing to keep the excitatory signals in check. Your neurons can start firing uncontrollably. Overexcitation. That can manifest as anxiety, confusion, and in severe cases, seizures.
It's terrifying that these mechanisms are so distinct, hitting a wall, hitting a ribosome, hitting DNA, but they all have these specific, unique, and dangerous ways of messing with the brain.
It just shows you how interconnected biology is. You pull a thread in a bacterium and a knot tightens in your brain. It's all chemistry.
And finally, group D metabolic pathway inhibitors. The source says these block folate synthesis.
Right, like trimethoprim, sulfamethoxazole. Bacteria need to make their own folate to build DNA. We get it from our diet, so that's another good asymmetry. These are often bacteriostatic.
Meaning they don't kill, they just stop the bacteria from growing.
Exactly. They put the brakes on replication, which is generally safer in terms of that explosion risk we talked about. But they have their own issues, often related to allergies and bone marrow.
Okay, so that's the bacterial battlefield.
Yeah.
It's already more complicated than I thought. But what about when the enemy is a virus? Section 3 covers antivirals. And the source makes a profound point. Viruses have few independent structures.
They are ghosts. You can't target a viral cell because there is no such thing. You can only target a process. You can't blow up their house because they're living in yours.
So what are the targets?
You can try to block entry stopping the virus from knocking on the door or picking the lock. You can block fusion, preventing it from merging with our cell membrane. Or you can attack the copying process.
Chain termination.
Exactly. You give the virus a faulty building block, the nucleoside analog, so when it tries to copy its genetic code, the chain just stops. The copy machine jams.
But there's a huge limitation listed here. Latent viruses are drug invisible.
This is the scary part. This is why we haven't cured things like herpes or HIV. If a virus decides to just sit there and sleep inside your cell's DNA, what we call latency, the drugs can't touch it.
Why not?
Because the drugs only work when the machinery is running. They work by jamming the copy machine. If the virus isn't trying to make copies, there's nothing for the drug to jam.
If it's not moving, you can't shoot it.
Exactly. And the CNS issue here is the blood-brain barrier, or BBB, which we've mentioned. Many antivirals are large, complex molecules. They aren't very fat-soluble. They bounce right off that protective barrier around the brain.
So the brain becomes a reservoir, a hiding place.
Correct. You might successfully clear the virus from the blood and the rest of the body, but it can hide out in the brain because the drugs can't get in at high enough concentrations. It's a sanctuary site. The virus just waits there, dormant, for the drugs to stop, and then it can reactivate.
That is deeply unsettling. Now let's talk about fungi again. We mentioned the toxicity because of the ergosterol-cholesterol similarity, but the source gets specific about the neural risks. It lists neurotoxicity, hallucinations, confusion.
Because fungal membranes and human neural membranes are so similar chemically, when you use a drug that punches holes in the fungal membrane to make it leak, you risk doing the same to your own neurons.
And that would disrupt their ability to fire properly.
Absolutely. It can destabilize the electrical potential across the neural membrane. There was a famous case with an antifungal called voriconazole causing vivid visual hallucinations, people seeing flashing lights and bizarre colors, because it was directly tweaking the neurons in the visual cortex.
I feel like we are seeing a theme here. The closer the evolutionary relationship, the messier the breakup.
That is a great way to put it. The more you look like the enemy, the more likely you are to get hit by friendly fire.
Which leads us to Section 4, antiparasitics. Parasites like protozoa and helmet swarms.
This is where the concept of collateral damage gets truly, truly dramatic. This is the scenario that keeps neurologists and infectious disease doctors awake at night.
The source mentions the critical risk of rapid parasite death. It's a paradox.
It is. So imagine you have a parasite living in someone's brain. A common one is neurocysticercosis, which is a tapeworm larva that forms a cyst.
A tapeworm in the brain. Okay, nightmare fuel established. I think we can move on.
It happens. It's actually one of the most common causes of adult-onset seizures worldwide. So the worm is sitting there, alive in a little cyst. But here's the crucial thing. The immune system is kind of ignoring it.
Why? Why would it ignore a worm in the brain?
Because the parasite is a master of disguise. It has evolved a cloaking device. It secretes molecules that actively suppress the immune response right around its little cyst. It creates a little bubble of, don't look at me.
Okay, so it's in stealth mode, hiding from my immune system.
You're the doctor. You see this cyst on the MRI. You want to give a drug to kill the worm.
Obviously. Get it out of there. That's the whole point.
So you give a strong anti-parasitic drug, like albendazole. The worm dies instantly.
Good, right. Mission
It's
accomplished.
terrible. Absolutely the worst thing that could happen in that moment. Because now you have a dead, rotting worm in your brain.
Yeah.
And critically, when the worm dies, its cloaking device fails. It stops secreting those immunosuppressive molecules. Suddenly, the immune system, which was blind before, can see, and it sees this massive foreign object that is dissolving and releasing thousands of antigens.
And the immune system wakes up.
It doesn't just wake up, it goes nuclear. You get a massive cytokine surge. The brain's immune cells swarm the area, causing intense inflammation. This leads to massive cerebral edema swelling and seizures.
So by killing the parasite, you actually triggered the very attack that damages the brain.
Exactly. You often see patients who are relatively stable with the live worm, but then they crash and have intractable seizures the day after you give the treatment. The treatment itself causes the emergency.
So the expert insight here is sometimes killing the parasite too fast is more dangerous than the parasite itself.
Absolutely. It's deeply counterintuitive. In many of these cases, you have to give high-dose steroids to suppress the immune system before you give the antiparasitic. You have to intentionally dampen the explosion before you light the fuse.
This brings us perfectly to Section 5. Resistance. We've all heard of antibiotic resistance. It's in the news all the time. But the source reframes it in a way I've never heard before. Resistance is information, not strength.
I love this definition. It's so much more accurate. We tend to anthropomorphize bacteria. We think, oh, the bacteria got stronger muscles. It have a thicker shell.
Right, like it's been working out in the gym. A Rocky Balboa bacteria.
It's not that. It's not about brawn. It's about brains. It's about data. It's about the bacteria learning a new piece of code that lets it survive.
What kind of tricks does it learn? What does this information do?
The source lists a few key mechanisms. One is enzyme destruction. The bacteria learns to build a specific molecular scissor, an enzyme like a beta-lactamase, that literally finds the antibiotic molecule and cuts it in half, rendering it useless.
So it disarms the bomb before it goes off.
Precisely. Another is target modification. It changes the shape of the lock, so our key doesn't fit anymore. It might alter the structure of its ribosome just enough that the antibiotic can't bind to it, but it can still make its own proteins.
And efflux pumps. That sounds interesting.
That is my favorite mechanism, purely from an engineering standpoint. It's basically a microscopic bilge pump. The antibiotic gets into the bacteria, enters the cell, and the bacteria has a dedicated pump that recognizes it as a toxin and just pumps it right back out before it can do any damage.
So it's not stronger, it's just smarter. It has better software.
It has better software. And here is the scariest part. That software, that genetic information for the pump or the scissor, is often stored on little mobile loops of DNA called plasmids. And bacteria can share these plasmids with each other.
Like trading cards.
Like airdropping a file. One resistant bacterium can cozy up to another bacterium, even one from a completely different species, and transfer the resistance file.
That is a terrifying image. Bacteria just airdropping resistance genes to each other in the hospital.
That is essentially what happens. It's called horizontal gene transfer. So you don't just have one resistant bug that has to reproduce. You have a network sharing the software update in real time.
Okay, we've talked a lot about killing the bugs.
Yeah.
But Section 6 introduces a fascinating twist. Immunomodulation. When killing isn't the answer.
We hinted at this with the parasites, right? Sometimes the severity of the disease isn't driven by the number of bugs. It's driven by your body's over-the-top reaction to the bug.
The host response.
Exactly. Your own immune system is causing the damage. In severe meningitis, for example, the inflammation can destroy the neural network much faster than the bacteria can. The swelling cuts off blood flow. The pressure itself kills neurons. The friendly fire is worse than the enemy.
So the goal shifts from kill, kill, kill to save the network.
Right. The priority is no longer just pathogen eradication, it's preservation of function. So we use tools like corticosteroids to suppress the overall inflammation, or more targeted drugs, cytokine blockers, to stop the storm.
The neurologic here is fascinating. It says, reducing inflammation can preserve cognition even if the pathogen is still there.
You are buying time. You are trying to keep the house from burning down while you deal with the intruder. If you kill the intruder but the house is a pile of ash, you haven't really won, have you?
This leads us to the therapeutic bottleneck, Section 7, the blood-brain barrier. We've mentioned it a few times, but let's really dive in. Why is it so hard to get drugs in there?
The BBB is an incredible piece of biological engineering. It's designed to keep almost everything out. It's the ultimate bouncer for the brain's VIP lounge.
How does it work?
The capillaries in the brain are different from anywhere else in the body. The cells that line them are stitched together with something called tight junctions. They are incredibly secure.
So nothing leaks through the cracks.
Nothing. To get past the bouncer, a drug needs to have the right credentials. It generally needs to be lipophilic, meaning it loves fat, so it can dissolve through the cell membranes. And it needs to be small. Most drugs are too big or too water-soluble.
But there's a paradox mentioned here. Inflammation actually makes the BBB more permeable.
Yes, this is a crucial point. When the brain is inflamed, like in meningitis, the whole system goes on high alert. The body needs to get immune cells into the brain to fight the infection, so those tight junctions loosen up a bit.
Bouncer gets distracted.
Or rather, the doors get deliberately propped open to let the good guys in. And when that happens, some of our drug molecules can sneak in with them.
So, weirdly, it is easier to get antibiotics into an inflamed brain than a healthy one.
Exactly. But here's the catch. As you treat the patient, they start to get better. The inflammation goes down.
And the doors close.
The barrier tightens back up. The bouncer is back on duty. Suddenly, your antibiotic might stop getting into the brain at the concentration you need. You might have a patient who is improving and then suddenly relapses a week later because the drug can no longer reach the few remaining bacteria. It's a constantly moving target.
That leads to the great debate in this section. Bactericidal versus bacteriostatic. To kill or to inhibit. This seems to be a huge topic in infectious disease circles.
It is the Coke versus Pepsi of our field, but, with life or death stakes.
So just to recap, bactericidal means it kills. It causes that 3-log reduction in bacteria. Beta-lactams, vancomycin.
Correct. These are the drugs that cause the lysis, the explosion we talked about.
But we established the risk. Rapid killing triggers the endotoxin release. The explosion causes swelling, which is terrible for the brain.
Right, so the argument goes, in bacterial meningitis, if you use a powerful bactericidal drug that shreds millions of bacteria instantly, you cause an acute, massive inflammatory burst. You might actually fry the brain with inflammation in your attempt to save it.
Okay, so the alternative is bacteriostatic, drugs like linezolid or tetracyclines.
These just stop the growth, they freeze the bacteria in place, they don't make them explode. This allows the host's own immune system to come in and clean up the mess slowly and carefully, like a controlled demolition rather than a bomb.
Slower controlled clearance, reduced inflammation. That sounds
I
better.
agree.
Why wouldn't you always do that in the brain?
Ideally, yes. In a perfect world, for a CNS infection, a bacteriostatic approach might be safer to prevent that inflammatory surge, but, and this is a massive but, it relies entirely on you.
Me?
It relies on your immune system. The drug just freezes the bug, but your own white blood cells have to do the actual killing and cleanup.
So if you are immunocompromised, if you're a chemo patient, for example,
If you are neutropenic, if you don't have enough of those key white blood cells, you can't use a bacteriostatic drug. It's useless. You are freezing the enemy, but you have no soldiers to go and capture them.
they'll just wait you out.
They will just wait you out. As soon as you stop the drug, they start growing again. In that case, you have no choice. You have to use the nuke.
You have to use the bactericidal drug, dissipate the inflammation risk.
You have to. It's a calculated gamble. You hope you can control the fallout with steroids.
The source mentions a clinical nuance, specifically for gram-negative meningitis. Why is that different?
Gram-negative bacteria are particularly nasty. Their outer membrane is loaded with something called lipopolysaccharide, or LPS, which is a very powerful endotoxin.
So when they pop, they release more poison.
A lot more. So rapid lysis from a bactericidal drug in a gram-negative infection is even more dangerous. It can cause a profound and transient worsening of the patient's condition. It's terrifying for a young doctor to witness.
How so?
You give the right drug,
Okay.
and 2 hours later, the patient's blood pressure plummets into shock, their fever spikes to 105, they become unresponsive. Your first instinct is, the drug isn't working. The infection is winning.
But it's actually the drug working too well.
It's the debris, the inflammatory response to the kill. You have to have nerves of steel and trust your training that this is a temporary phase caused by the treatment itself.
So what is the final verdict? Is one better than the other?
There is no absolute superiority. It's all about context. What's the bug? What's the state of the host's immune system? Where is the infection? That's the art of medicine.
Okay, let's move to section 8. And this is the part I think will resonate most with our listeners. We've been talking about severe brain infections, but this section is about collateral damage that costs to the host, in a broader sense, even for routine stuff.
This is huge. We often think of antibiotics as having no biological cost to us other than maybe the price at the pharmacy. We think they are free, but the biological cost is high. They disrupt host homeostasis in profound ways.
And the source breaks it down into 3 axes. Damage axis 1, microbiota disruption, dysbiosis.
We all know this one on some level. You take a broad-spectrum antibiotic. It's like carpet bombing your insides. You might kill the pathogen in your sinus, but you also wipe out the trillions of beneficial bacteria living in your gut garden.
And we know about the gut-brain axis. We've done entire deep dives on this.
We have, and it's so important here. The bacteria in your gut produce short-chain fatty acids that feed your colon cells. They produce precursors to neurotransmitters like serotonin and dopamine. They communicate directly with your brain via the vagus nerve.
So when you wipe them out with an antibiotic,
That entire signal line gets cut or distorted. The chemical conversation between your gut and your brain is disrupted.
and the result, according to the source, is neuropsychiatric effects like anxiety and depression-like states.
Literally, the death of your gut bacteria can make you feel anxious or depressed. There are fascinating studies showing that mice given broad-spectrum antibiotics show behavioral changes that look exactly like anxiety. They become more timid, they explore less.
Damage axis 2, we touched on this with the ribosomes, but let's go deeper. Mitochondrial toxicity. The source calls antibiotics mitochondrial toxins. That's a strong phrase.
It is, but it's accurate. Because of that endosymbiotic origin we discussed, many antibiotics are, by their very nature, toxic to our mitochondria. When you disrupt the bacteria, you risk disrupting your own cellular power plants.
The mechanism is listed as oxidative stress and impaired ATP production. What does that mean in plain English?
It means energy failure. Your mitochondria are like little engines that burn fuel to produce ATP, which is the energy currency of the cell.